Before I go on, are there any questions on any of the stuff that we've covered so far? All right, I think we left off, we had discussed the structure of IgM, and now we want to talk about some of the major biological properties and functions of IgM. IgM is the third highest concentration in serum, so IgG is first. I ordered these based on somewhat on their structure rather than their serum concentration. So IgM is the highest, IgG is the highest, IgM is the third most common immunoglobulin in the serum. It is the first immunoglobulin that we're able to make. As fetuses, we actually begin making, we begin to get immunocompetent, and we begin making immunoglobulin, and we start making IgM. We don't become fully immunocompetent until after birth, however. So it's the first one that is made, that we're able to make as a fetus. It is also the first immunoglobulin that the B cell is able to make. So that when the B cell is going to be activated by antigen, the very first things it's going to be able to make is IgM. And when we get to the genetics, we'll see why that's the case. It is a complement-fixing antibody, so it will fix complement and mediate that effector function. In fact, IgM is actually, turns out, is a better complement-fixing antibody than is IgG. And let me illustrate why. If I can get it to go to the next slide. There we go. Okay. If you'll remember, if you think back to the complement lecture, I showed, when we were talking about the classical pathway, the very first pathway, I showed you that the C1q was cross-linked by two immunoglobulin molecules. I had two immunoglobulin molecules there, and C1q was cross-linked by those two. That wasn't an accident. The reason is that in order for complement to be fixed, the C1q must be cross-linked by two immunoglobulin molecules. So in the case of IgG, now IgG is a complement-fixing antibody, but if you had a situation like this where you had two IgG molecules, but they were too far apart so that they couldn't cross-link C1q, it would not be fixed, and you would not start that complement cascade, all right? So only when the two molecules are close enough together that they can cross-link C1q will you get activation of C1q with an IgG antibody. But in the case of IgM, since it is a pentamer, even a single molecule of IgM is able to cross-link C1q because it's got this pentameric structure. So IgM turns out to be a better complement-fixing antibody than even IgG is. Now, there is some practical importance to that, and that is that I just told you that IgM is the first immunoglobulin to be made in an immune response. One of the things that we do in staging the stage of an infection, if you have a patient that has an infection, you're trying to stage where it might be in the infection. Is this a new infection current, or is it maybe an older infection, or was this person infected in the past? One of the things that's done is to look for antibodies to IgM. If you're seeing IgM antibodies, that's telling you that it's early in the response. This is the beginning of the response. If you see mainly IgG antibodies to the antigen, then you're either talking about late in a current infection, or you're talking about some prior infection that gave rise to those IgG antibodies. All right, so you can use the class of immunoglobulin to help you stage the stage of the infection. Well, one of the ways that they then determine whether or not it's an IgM antibody is to look to see if they're good complement-fixing antibodies. So if they're good complement-fixing antibodies, that's a suggestion that you're dealing with an IgM antibody and that this is some early stage in the infection process. So we take advantage of these little features of the immunoglobulins to actually give you some very important information. And so you're going to be hearing, and when you get into the bacteriology and virology section of the course, that there's a complement fixation test for this thing. They're looking for IgM antibodies in those cases. Realize that then it's not foolproof. I mean, IgG will fix complement. It's just that it's not quite as good a complement-fixing antibody as is IgM. Okay, IgM also is a very good agglutinating antibody, and agglutination just simply refers to the aggregation of a particulate antigen. So if you have a bacterium, for example, which is a particulate antigen, IgM is very effective in agglutinating and clumping these. And again, it's a consequence of its structure. Its valence, at least theoretically, the valence is 10. So each molecule of IgM can aggregate up to 10, theoretically at least, 10 bacterial particles. So it's an extremely good agglutinating antibody. There are some cells that have receptors for Fc receptors for IgM, and then the IgM can help modulate the activity of those cells. But it's not very, very common. A major place that IgM plays a major role is it is one of the immunoglobulins that is on the surface of B cells. B cells have two classes of immunoglobulin. Mature B cells, prior to exposure to antigen, have two immunoglobulins on their surface. One of them is IgM. And the role of that IgM is to serve as the receptor for antigen. That's how B cells recognize antigen, because they display on their surfaces IgM. We'll see what the other class is shortly. Now, when IgM exists on the surface of the B cell, it does not exist in that pentameric form. It exists as a monomer. And it actually has an extra little stretch of amino acids at its C-terminal end, at the end of the Fc region. And it's only about 20 or 30 amino acids long, but there's very hydrophobic amino acids in, and that allows the IgM to be inserted across the hydrophobic membrane. So this becomes a transmembrane protein because of this tailpiece allows it to insert into the membrane. All right, and as I said, this then acts as the receptor for antigen. When that B cell encounters antigen binds to the IgM that's sitting on the surface of the B cell, and that's one of the signals that B cells need in order to become activated to go on to differentiate into immunoglobulins, or into plasma cells which secrete immunoglobulin. The immunoglobulin that's going to be secreted by the B cell that gets activated has the same specificity as the IgM that was on the surface of that cell. All right. Now, when IgM exists on the surface of the cell, the actual B cell receptor is a little bit more complicated. It is the IgM molecule, but there are some proteins that are associated with it, Ig alpha and Ig beta, they're called. And these proteins play an important role in the transmission of the signal. Because you see, the way that when IgM inserts into the membrane, if this is the membrane here, when IgM inserts into the membrane, that tailpiece does extend into the cytoplasm, but it's just a few amino acids that are in the cytoplasm of the cell, and it's not that is not long enough to transmit a signal and start a cascade of events. So the IgM associates with these accessory proteins, Ig alpha and Ig beta. And then what happens? When antigen binds up here, conformational changes occur, and then the signal is transmitted actually through these additional proteins. And I'm not going to go into what those signal transduction pathways are, but suffice it to say they involve phosphorylation of proteins and a whole series of secondary messengers. Ultimately, what happens is we get the activation of a number of genes that allow the cell, that kick the cell back into cell cycles, so they begin to divide and expand that clone of cells. The immunoglobulin begins to be expressed and ultimately secreted. So we activate the immunoglobulin genes, make a lot of immunoglobulin, and then the cell secretes that. So these accessory proteins are important for transmitting that signal. Yes?
Antigen has to be bound to it? Or?
Yes, there's no signal is going to be, when antigen binds up here, that's one of the signals. Now, remember, we've got T-dependent and T-independent antigens, so they have to be handled differently. But in the case of T-dependent antigens, one of the signals is going to be antigen binding up here. A second signal is going to have to come from the T cells. Okay? In the case of the T-independent antigen binding here is going to be enough to send that signal. Okay? And so we get a lot of secondary messages that go through, and you ultimately activate these genes. They kick the cell into its final stages of differentiation. Okay. IgA. As far as the structure is concerned, in the serum, The predominant form of IgA in the serum is the monomeric form. There are some aggregates, larger components of IgA, but predominantly, it's like 70 or 80% of it is monomeric. But in the secretions, where IgA plays its major role, IgA exists as a dimer. And in the secretions, it's referred to as SIgA for secretory IgA. Okay? And so it exists as a dimer. Again, the older nomenclature, you'll see it sometimes referred to as 11S immunoglobulin. Again, as a polymerized form, it has the J chain associated with it because all the polymerized forms, like IgM, have the J chain associated with it and is involved in that polymerization. Now, IgA also has, secretory IgA, has an extra piece that is indicated here in this cartoon as by this goldish wavy line. That is called the secretory piece or secretory component. Sometimes it's referred to as the T piece. This is an interesting protein because it turns out that protein is not made by the B cells. B cells make everything else. B cells make the heavy chains, the light chains. They assemble the immunoglobulin. They make the J chain. The B cell makes the dimer, their dimeric form, and then secretes a dimeric form of the IgA from the cell. The secretory piece is not made by the B cell. The secretory piece is actually made by epithelial cells and is picked up by the immunoglobulin as the immunoglobulin is being transported across the epithelial cell barrier. And let me show you what happens here. This is an epithelial cell barrier. As you're going to be looking at it, the lumen is going to be to your right. This is the adluminal side. Okay? So on the adluminal side of the epithelial cells, there is a receptor for immunoglobulin. Now, this receptor is actually a receptor for polymerized forms of immunoglobulin. So it actually binds both IgA and IgM. However, the affinity of that receptor for IgA is much, much larger than the affinity for IgM. So it's primarily a receptor for IgA for all practical purposes. If we're immunologically normal and making both IgM and IgA, this thing is preferentially going to bind IgA. In individuals that are IgA deficient, there are certain IgA deficiencies. In those individuals, IgM will bind to this particular receptor. But in a normal circumstance, it's essentially an IgA binding protein. Okay? So as the B cells make and secrete the IgA binds to that receptor. The binding of the receptor results in the internalization of the IgA and the complex in vesicles, and those vesicles then are transported across the epithelial cell where they fuse with the luminal surface and release the IgA. But what happens in this acidic environment in these vesicles, this bond here is actually, it's proteolytically digested actually, to release part of the receptor in association with the IgA. That is the secretory piece. So the secretory piece is actually part of the receptor that is used to transport the IgA across the epithelial cell barrier so that we get IgA in the secretions. So that secretory piece plays an important role in that transport. It is actually part of the receptor. The other role that the secretory piece plays is that it protects the IgA from degradation by proteases in the secretions. There's a lot of there's a lot of proteases out in these secretions, and the IgA would be susceptible to digestion with these proteases, except that it's protected by the secretory piece. Once you remove that secretory piece, the IgA is rapidly destroyed. So it really protects the IgA in the secretions. Okay, so what are some of its properties? It's the second highest concentration in serum. So it's IgG, IgA, IgM. That's the order of the serum concentration. It, however, is the major immunoglobulin that you find in secretions because of this ability to be transported across the epithelial cell barrier. And therefore, the major role that IgA plays is in what is referred to as the mucosal or local immunity. It's at the site of where we are going to get infected in the secretions. So we find in the tears, in saliva, the gastric secretions, in the pulmonary secretions, you find lots of IgA, and it's there to block any infections from even taking hold. All right, so it plays a role in what is referred to as this local immunity or mucosal immunity. So it's a very important immunoglobulin in that sense. It's not a complement-fixing antibody, so it doesn't mediate any of those functions. Now, you notice I put in here parentheses, unless aggregated. I have seen in some of these board review books, and actually I think years ago I actually even saw it on one of the board exams, they asked this question that dealt with complement fixation, and one of the foils in there was aggregated IgA, and the idea was that you had to know that aggregated IgA does fix complement. Well, in fact, it does, but it doesn't make any, I mean, that's a laboratory artifact. We don't have aggregated IgA floating around in our bodies. So as far as I'm concerned, you don't have to remember that. If you want to remember that little minor fact, go right ahead, but I don't care. It's, as far as I'm concerned, it's a not complement-fixing antibody, okay? There are some cells, however, that have receptors for IgA, so IgA can help modulate the activity of those cells. Okay. IgD. IgD exists as a monomer, and so nothing special about its structure, except that it's got this extra little tail piece, just like IgM has, because IgD is the other class of immunoglobulin that you find on mature B cells. All right? It is the fourth highest in serum concentration. So it's IgG, IgA, IgM, and then IgD. But there is a huge difference between the amount of IgM and IgD. So for example, IgM is roughly there in about 0.5 milligram per mil, something like that, per mil of serum. When you get to IgG, we're talking about microgram quantity. So we're talking about 1,000-fold less IgD than IgM. All right? In fact, it's not even clear if IgD plays any role in the serum. It's thought that that low concentration of IgD in the serum is probably there because it gets sloughed off of B cells. Because where IgD is important, as I said, is it is the other class of immunoglobulin that is on the surface of B cells that acts as the antigen receptor. So mature B cells have on their surface two immunoglobulins, IgM and IgD. And those antigens, or those immunoglobulins, act as the antigen receptor. All right? And when IgD is on the surface of the cell, just like IgM, it associates with that IgA, Ig alpha, and Ig beta, and the same story is true. Now, although the B cells have on their surface two classes of immunoglobulin, IgM and IgD, the specificity of the antibody, in other words, the antigen that it's going to bind, is going to be the same. So the IgM and the specificity of the IgM and the specificity of the IgD are identical. You have the same, they bind the same antigen, all right? And we'll see again when we get to the genetics why that happens. And finally, it's not a complement-fixing antibody, and there are no cells that have receptors for IgD on their FC receptors for IgD. IgD, it seems the main role of IgD is basically to act as the antigen receptor on B cells, and it probably plays no role in the serum. The serum stuff is probably there because these B cells may slough off a little bit. All right, IgE. Structurally, again, it's a monomer, but it's one of these classes that has an extra domain, so if you count them, CH1, CH2, CH3, CH4, or in this case it's an epsilon 4, but since it's an epsilon chain. So it's the only thing structurally is it does have an extra domain. As far as its properties, it's actually present in the serum in the lowest concentration. So the order is G, A, M, D, E. So E is there at very, very low concentrations, but the reason it's there at very low concentrations is different from what I just described for D. In the case of IgE is actually secreted into the serum, but the basophils and mast cells, tissue mast cells and basophils, have a very high affinity receptor for IgE. So as soon as IgE is made and secreted, it is essentially it's absorbed onto the surfaces of the basophils and mast cells. Again, this is one of the exceptions. This absorption onto that FC receptor is not dependent upon antigen. So the IgE, even before it is exposed to antigen, will bind to basophils and mast cells. So the reason that IgE is there in such low concentration is that as soon as it's made, it's basically sopped up by all the basophils and mast cells, and it's displayed on the surface of these cells, because those cells have a high affinity Fc receptor for IgE. Now, IgE plays a major role then in allergic reactions, because what happens in allergy is that when we are exposed to the allergen, the antigen to which we are allergic, that allergen then binds to the IgE on the basophil and mast cell and causes the basophil and mast cell to degranulate. And those granules contain all these pharmacologically active materials that give us the symptoms of allergy. Right, you're going to have a whole lecture on allergy, and you'll see, but IgE plays a central role in these allergic reactions. IgE also plays a very important role in parasitic infections, particularly the worm infections, which are what the helminths are. These are worms. And IgE is important in worm infections because the eosinophils have an Fc receptor for IgE as well. And the IgE binds to the Fc receptor on eosinophils, causing them to degranulate. And when the granules from the eosinophils are particularly helpful in killing worm parasites. And so the IgE plays a major role in these types of infections. And what you see, for example, one of the things that you can do, you'll do clinically, if you're dealing with a patient with an allergy or dealing with patients with worm infections, one of the things that you can do is to look for increased levels of IgE in the serum, because if we're exposed to these, our IgE levels start going up. So monitoring IgE levels is one thing that you can do in helping diagnose worm infections or allergic reactions or allergies. It's not a complement-fixing antibody, so it doesn't mediate any of that particular effect or function. All right. Now, I have included in your handout I think it's two-page list of a whole series of diseases. We're looking at immunoglobulin levels, either where they go up or they go down, where they might be helpful. That is not there to be memorized, all right? I just gave it to you just to give you an idea that there are a lot of different areas where you're going to look for immunoglobulin levels and to help you in making diagnoses. So just look through them. Get a feel for the fact that there are a number of places that these things are going to be helpful to look at. But I'm not going to ask you about any of those particular diseases, okay? All right, any questions on the immunoglobulins? Anything? All right. So we'll go on and now discuss the immunoglobulin isotypes, allotypes, and idiotypes. All right. Now we're going to take a change of direction here. So far, we have been talking about the immunoglobulins as antibodies. And certainly that's what they are. That's their function. They function as antibodies. But an immunoglobulin is a protein, right? Basically, it's a protein. I mean, it does have some carbohydrate on there, but it's basically a protein. And so there's no reason why we can't use that protein as if it were an antigen. In other words, there's going to be some antigenic determinants on an immunoglobulin molecule that theoretically we can make antibodies against. So in this discussion of isotypes, allotypes, and idiotypes, we have to think of the immunoglobulin now in terms of the fact that it is an antigen that we're using to make antibodies against, and we're going to discuss the various types of antigenic determinants that you find on an immunoglobulin molecule. And the determinants that you find on the immunoglobulin molecules are called isotypic determinants, allotypic determinants, or idiotypic determinants. All right? And we're going to discuss each of those. So keep in mind we're dealing with the immunoglobulin as an antigen. Okay? And we'll start with the isotypes. So what are isotypes? Here's a nice dictionary definition. Antigenic determinants that characterize the classes and subclasses of heavy chains and types and subtypes of light chains. Okay? So what does that mean? Right? Now, if everybody would just close their, or certainly don't look at your laptops, all right? I want to just go through this little exercise, and if I see you looking at your laptop, you're the one I'm going to ask the questions to, okay? And you may know the answer to the question, but then I'm going to ask you why, okay? All right. Suppose we do this. Suppose we take some human IgM, and I inject that human IgM into a rabbit. Well, obviously, this is a protein, so this rabbit might recognize things on there that are going to be antigenic determinants. In the end, the rabbit's going to respond to those and start making antibodies. So we could take the serum out of that rabbit, and we'll refer to that as an antiserum because it's a serum that contains antibodies against whatever we use to immunize with. So the question is, what will that antiserum react with? Do you think it will react with IgM, human IgM? Sure. I mean, that's what we immunized with, right? So it'll react with human IgM. Will it react with human IgG? That becomes a little bit more difficult, huh? Will it react with human IgG? Okay, we got a yes. We got a yes. Do we have everybody agree? It's a yes? Okay, we got to have a dissenter. Anybody else dissent? How many just don't know? Okay. Okay, we'll have yes, no, and abstain, right? All right. Somebody that said yes. Why did you say yes? Somebody want to volunteer?
Those two kind of go together in my mind.
Okay. Anybody else got an idea why? Besides just intuition. If they're variable regions.
If they both have the same specificity.
That's possible. But it's not likely that, though, you're going to have, remember when I take an IgM, I'm just taking a whole population of molecules. They have lots of different, specificities. So that's possible, but probably not likely.
Do you have any other things that would react with general antigens with foreign bodies? So would it develop?
I'm not sure what you're asking. I mean, we just simply took this IgM, injected it in, and the question is it going to react with IgG? Some people say yes. I told you yesterday when I said, what happens if, when we were talking about immunoglobulins and I said, this is IgG. What did I tell you? I told you that it's going to have everything there. So when I inject IgM, at the very least, I am also injecting kappa light chains and lambda 1 and lambda 2. Those are the same things that are present on IgG. So at the very least, when this animal recognizes this, anything on any anti-light chain activity that's present in this serum is going to react with IgG as well. This will react with all immunoglobulins, right? It'll react with everything. Because at the very least, there's the light chains in there stimulate the production of the antibodies, and those same light chains are on all these different classes of immunoglobulins. I told you. Okay? So it's going to react with everything. All right. Now, suppose I take this this antiserum, and I react it with IgG, IgA, IgD, and IgE. Right? And again, without me telling you how I can do this, but let's say that there is some way that after I let the antigen antibody complexes form, I can remove those things. So I can remove all the immune complexes that form with these, and then I have what is referred to as an absorbed antiserum. It's been absorbed. And so now the question is, what's this going to react with? Will it react with IgG? No, because now anything that could have reacted with IgG would have been reacted with this and has been removed. So all those antibodies to light chains and kappa light chains, all that's going to be removed. All right? So this is not going to react with IgG anymore. Will it react with any of these classes of immunoglobulins? Will it react with IgM? Yes, it's still going to react with IgM. Now, it's going to react only with IgM. So this is now specific for IgM. What's it reacting with? What do you think it's reacting with? What makes IgM IgM? The heavy chain constant region. So any determinants that are present in the heavy chain constant region that are unique to the mu heavy chain is going to be what this is reacting with. So this is reacting with determinants that are specific for the mu heavy chain. Those determinants that are unique to that heavy chain are referred to as isotypes. Now, I can go through the same process. I can immunize with IgG here and come down here and then absorb, change this to M, and I could produce an antiserum that is specific for G. All right, the gamma heavy chain, or the gamma 1 heavy chain if I want to do it that way, or gamma 2. So each class and subclass of immunoglobulin has its own unique set of antigenic determinants that are specific for that particular heavy chain. Those are the isotypes. And I can do the same exercise with light chains. I can inject kappa light chains up here, absorb with lambda light chains, and I could produce an antiserum that recognizes specific determinants that are only found on kappa light chains, or only found on lambda light chains, or lambda 1, lambda 2, lambda 3. So each class and subclass of immunoglobulin has its own unique set of antigenic determinants that are unique to that class or subclass of heavy chain, type or subtype of light chain. And those are called isotypic determinants, or the isotypes. All right. Is that clear what they are? All right. So where are they located? Well, by definition, what makes a mu chain is the heavy chain, right? So the isotypic determinants are going to be localized to the constant region of the heavy chain, because that's what makes it a mu. In the case of the light chain isotypes, it is located on the constant region of the light chain, because that's what determines the type of light chain, is the constant region. All right? So they localize to the constant region of the heavy chain or the constant region of the light chain. Now, you'll notice that in this particular diagram, I put in this part here is, of course, corresponds to CH1, right? This is the first domain of the constant region. You notice I put it in gray. And the reason for that is, I mean, theoretically, there are isotypic determinants in this portion of the molecule as well. In practice, when you do the type of immunization that I just described, where you inject the whole molecule, typically you don't produce antibodies against that part of the molecule. The antibodies are against this part of the molecule or against this part of the light chain. So, I mean, theoretically, they should be there, but to get them, to identify those isotypes, you have to do things a little bit differently than just the way we described it. All right, so let's take a look at a situation. We have an Ig1 kappa and an IgM kappa. All right, these two immunoglobulins will have different heavy chain isotypes, right? Because this is a gamma 1, this is a mu, so they have different heavy chain isotypes, but they have the same light chain isotypes, right? Because they both have kappa chains. All right, so where do we find the isotypes? What are the occurrence of these? The isotypes are found in all immunologically normal individuals. Okay, so every one of us, if we don't have some sort of immunodeficiency disease, every one of us expresses all of the isotypes. We all have IgM, so we have all the IgM isotypes. We all have IgG1, IgG2, we all have IgA, we have all the classes. Similarly, we all have kappa and lambda chains, so every one of us expresses all of these isotypes. Now, in fact, that's what the term iso means. The term iso comes from the Greek meaning the same among members of the species. So every one of us has all of these isotypes if we're immunologically normal. All right. So what are they used for? Well, the Isotypes are what is used to identify and measure immunoglobulin levels. When you go and you're going to send a patient sample down and you want an immunoglobulin level down, they're not going to go and isolate the immunoglobulin and sequence it to try to determine what constant regions the immunoglobulin has. I mean, that is a huge task. So that's not what a clinical lab does. What the lab is going to do is they're going to go over to the shelf, and there's a series of reagents. This antibody will be specific for IgM. This antibody will be specific for IgG1. And they use those reagents then to evaluate the immunoglobulin levels of each particular class or subclass of immunoglobulin that you may request that you want to get information on. So the isotypes are what the clinical labs use, basically, to measure immunoglobulin levels. And when we talk about antibody reactions, and we'll go over some of the tests that are used in some of the labs for doing these kind of things. These things can also be helpful. Detecting isotypes can also be helpful in helping diagnose B-cell tumors. Remember I said B cells have on their surface immunoglobulin. So they have certain isotypes expressed on that immunoglobulin that's on the surface of the cell. Now, if we look in humans, if you look at B cells and you ask what proportion of the B cells express kappa chains and what proportion of the B cells express lambda chains, it's roughly 50-50. It's different in other species, but in humans it's roughly 50-50. So half of our B cells, if I were to take a sample of your cells and look at them, whether they have kappa or lambda, half of them will have kappa, half of them will have lambda. Now, if you have a B cell tumor, though, you get a malignant proliferation of a clone of cells. So one clone of cells starts proliferating, and you have a lot of B cells then, all of which are derived from one clone. So they will have just either a kappa or a lambda, right, on them. So by looking and counting the number of B cells that express kappa and lambda isotypes, if you find in your patient that there's a preponderance of kappa, you know that there's something wrong. It's supposed to be roughly 50-50. So if 90% of your cells are expressing kappa, that's an indication that you're dealing with a B cell tumor that's expressing a kappa light chain on its surface. All right. So this can be helpful in looking at B cell tumors. Immunodeficiencies. I mean, this is how one of the ways that you help diagnose the immunodeficiencies. What's going to happen if a person has an IgA deficiency? He's not going to have any IgA in his serum. You take some of his serum and you measure the amount of IgA that's in that serum, and you'll see that it's lower than the normal range. Okay, so these isotypes are what are commonly used in the labs, basically, to identify all these different classes and subclasses of immunoglobulins in the types and subtypes of light chains. So they're very important, actually. They're used a lot. All right. Any questions on isotypes? All right. Then let's go on to allotypes. And again, here's our nice dictionary definition. They're antigenic determinants specified by allelic forms of the immunoglobulin genes. All right, so what does that mean? I like to do this one by analogy. Every one of us in this room has hemoglobin. I hope. Okay, so we all have hemoglobin. But it doesn't mean that every one of us has exactly the same hemoglobin. Most of us probably have hemoglobin A, which is the hemoglobin associated with adult hemoglobin. But there may be some in this class that have hemoglobin S, the hemoglobin associated with sickle cell anemia, sickle cell trait. Do you know what the difference is between normal hemoglobin A and hemoglobin S? 1 amino acid difference, all right, in I think it's the beta chain of hemoglobin. So it's just 1 amino acid substitution, okay? So just as in the case of hemoglobin, where you can have everybody having hemoglobin but being slight differences, the same thing applies in the immunoglobulin molecules. Every one of us has got IgG. But that doesn't mean that your IgG is exactly the same as my IgG. I may have inherited a gene that specifies a particular amino acid at this position in the chain. You may have inherited a gene for the immunoglobulin that specifies a different amino acid. And therefore, if I were to take my immunoglobulin and inject it into you, and take your immunoglobulin, or IgG, and inject it into me, the possibility exists that I would respond and recognize your immunoglobulin as being different. That one amino acid difference may have created a new epitope that I would recognize as foreign. I would make antibodies against it. You could make antibodies against my immunoglobulin. All right? Those differences are the allotypes. Okay? So differences that are specified by different allelic forms of the immunoglobulin genes can give rise to these allotypes. All right, so where do we get and how do we identify the allotypes? Obviously, the only way that you know an allotype exists is if you have an antibody to detect the allotype, all right? Now, we don't normally go about taking my immunoglobulin and injecting it into you or your immunoglobulin and injecting it into me. We don't normally do those things, all right? But there are instances where, in fact, we do that. And one of the places is in blood transfusions. If you've ever had a blood transfusion, they will match you for blood group antigens, but they don't match for immunoglobulin allotypes to make sure that you have the same immunoglobulin allotype. And the reason that it's not matched for immunoglobulin allotypes is that clinically it doesn't make any difference if you inject a person with a different allotype immunoglobulin. They'll just make antibodies against it, okay? It's not a problem clinically, so we don't do it. So anybody that's received a blood transfusion potentially is a source of antibodies that would be directed against the allotype of whatever the donor had, right? The donor allotypes. Another place that we get these antibodies is from women that after they've given birth. At birth, there's a rupturing of the membranes, and there's a mixing of fetal and maternal circulation, all right? And so what happens? If the child has inherited an allotype from the father that the mother doesn't have, the mother will essentially become immunized against that allotype. So women then become a source of these anti-allotypic antibodies that are used to detect the allotypes. All right, so where are they found? Theoretically, there's no reason why the allotypes couldn't be anywhere on the immunoglobulin molecule. In theory, they should be everywhere. But in fact, all the known human allotypes are located to either the constant region of the heavy chain or the constant region of the light chain. Again, there's no reason why that has to be the case. In fact, for example, in rabbits, there are variable chain allotypes in rabbits. So, I mean, it's possible. But for some reason, at least all the known ones in humans, that doesn't mean that there aren't some unknown ones, but all the known allotypes in humans are located to the constant region of the heavy or the constant region of the light. So let's look at a situation. We have here two IgG1 kappas from two different people, okay? These are both IgG1 kappas, so they share the gamma-1 isotypes, which are indicated in yellow here. You have the same gamma-1 isotypes. You have the same kappa isotypes because the isotypes are the same. But they have different allotypes shown in orange because they're from different people. This one has one set of allotypes. This one has a different set of allotypes. On both the heavy chain and on the light chain, you can have the allotypic differences as well. Okay? So the allotypes are located then in these constant regions. So where do they occur? Well, obviously, each of us has his or her own unique set of allotypes. All right? Now, some of us may share certain allotypes, that's true, but whatever genes you've inherited will determine what allotypes you have. Whatever genes I've inherited determine my allotypes. Okay? So the term allo comes from the Greek, meaning different among members of this species. So there are differences. Each of us may have some differences from each other. Now, actually, let's take a break now, and then I don't want to introduce the usefulness of these allotypes come from an understanding of the genetics, and that's what we'll cover next. So let's take our break now, and then we'll pick up here. Okay, can we get started? Is it okay? All right. As I said, the usefulness of the allotypes and what their value is to us is really comes from an understanding of the genetics of the allotypes. So what I want to do is I want to just talk to you a little bit about some of the human allotypes, human immunoglobulin allotypes, and then about their genetics and then how they're used. All right, first of all, just as far as the nomenclature is concerned, the nomenclature really gives you everything that you need to know about the particular allotype. All the immunoglobulins the allotypes are indicated with the nomenclature like this. The first part of the name tells you what chain it's on. So obviously this is on the gamma 1 chain. So this is going to be an allotype on the gamma 1 chain. The M is the symbol that this is an allotypic marker. So this is an allotypic marker on the gamma 1 chain. All right? And then following that, in parentheses, is some number, and that tells you which allotype you're dealing with. So this is allotype 3 on gamma 1 heavy chains. This is allotype 1 on kappa light chains. All right? So the name will tell you what chain it's on, and it'll tell you which allotype it is. And here's an example of some allotypes. So for example, by the way, you may see in some older texts, instead of a number in the parentheses, they'll have a letter. That is an older nomenclature, and if you see it, don't get concerned about it. It just they use letters instead of numbers. So the allotype 3 is actually in the old nomenclature was allotype F. All right, but what allotype 3 is, G1M3 is actually an arginine at position 214 in the chain. So if you have a gene that specifies arginine at position 214, then you will express allotype 3. You will be G1M3. All right. In contrast, if you have a gene that specifies lysine at that position, then you will be G1M17. All right. So you will have that allotype. Some of the allotypes are a little bit more complex. It's more than a single amino acid. G1M1 is actually this sequence of amino acids, arginine, asparagine, glutamate, leucine, at these positions, okay? So if you have a gene that specifies those amino acids at that position, then you will express that particular allotype. And you have allotypes on the light chains, like the KM allotypes. The KM1 is valine leucine at those positions. And KM3 is alanine valine at those same positions, all right? So depending upon what gene you've inherited, that will determine which allotypes that you have. Now, the genetics is these allotypes like the KM allotypes and the G1M allotypes like 3 and 17 and KM1 and KM3, see, they represent alternate amino acids at the same positions in the chain, all right? Those types of allotypes are inherited as codominant autosomal genes, so that neither one is dominant over the other. They both are expressed, so that if you are an individual that is a heterozygote, KM1, KM3, for example, heterozygote, you will be, if I were to take some of your immunoglobulin and look for the presence of the allotypes, I would find both 1 and 3 expressed, all right? And they follow just simple Mendelian genetics. If we have a heterozygote crossed with a homozygote KM1, roughly half of the offspring will be KM1, and the other half will be heterozygote. Okay, so it's simple Mendelian genetics. Now, although these are expressed as codominant alleles, so that in a heterozygote, both the KM1 and the KM3 are expressed, if you look at any individual and immunoglobulin molecule, all right, so now just look at one molecule. Each molecule would be either KM1 or KM3, but never both. Because remember, an immunoglobulin molecule, they're composed of 2 identical light chains. There's no differences between them. All right, so in any individual molecule, we'll have either KM1 or KM3, but never both. But in the population of molecules, some will be KM1, some will be KM3. Now, the reason that happens, that individual molecules only express one or the other, is that there is a phenomenon called allelic exclusions. And what that means is that B cells exclude one allele. When they express the immunoglobulin genes, they exclude one of the alleles. Remember, we have 2 alleles for every, two alleles for all of these genes, because we've inherited one from our mother, one from our father. All right? So the B cells exclude one. It's called allelic exclusion. And therefore, the B cell only makes, either are the expresses the gene from the mother or the gene from the father, never both. And that's why the immunoglobulin secreted by that cell only has identical light chains and expresses only one light type, or one kappa light chain allotype. All right? Is that clear? Yes.
Going back to the million, it'll be about 50-50.
They're roughly 50-50. Right. So about half the molecules will be KM1, half the molecules will be KM3. All right. All right. Now, it is possible, however, to have more than one allotype on the same chain. So, for example, if you have a chain like, if you have an IgG1 like this, at position 214, if you express lysine, and at positions 355 to 358, you express this set of 4 amino acids, then obviously that particular chain will have two allotypes. It'll have 17 on it and 1 on it. Okay? So it is possible in these situations where they're not, where the allotype does not represent different amino acids at the exact same position, then it is possible to have two allotypes on the same chain. But it would never be possible to have an arginine and a lysine at the same position in an individual chain, right? You have to have one or the other. So in these cases, you do get on one chain, you may get two immunoglobulin allotypes expressed. But in the case of the KMs or GM1, GM3 and GM17, that could never happen. You'd have to have one molecule could express 3, one molecule could express 17, but you could never have a molecule that expresses both in those instances. All right. So what are the allotypes used for? Well, there are a number of practical applications of the use of allotypes. One is in monitoring bone marrow grafts. What do you do in a bone marrow graft? In the recipients, for whatever reason, the bone marrow, if it's either not functional or has been destroyed because you're trying to deal with a tumor, you get rid of the recipient's bone marrow, basically, by irradiation or by chemical treatment. And you're going to repopulate the bone marrow with a transplant from the donor. Now, again, they will match the donor and the recipient for all sorts of things, all right? For blood group antigens, they're matched for their blood, they're matched for tissue type, and then so on, as close as they can get the match. And then they do the transplant. But they don't match to see whether or not you have the same immunoglobulin allotypes. And the reason is, again, because clinically it doesn't make any difference. There's no problems clinically if you receive a bone marrow graft from a donor with a different allotype. It doesn't make any difference. So it's not matched. But we can make use of that then in finding out whether or not that bone marrow takes. If the bone marrow is taking, what's going to happen? The bone marrow is going to repopulate the bone marrow of the recipient. Those cells, the stem cells in that bone marrow are going to begin to differentiate into lymphoid cells, and some of those lymphoid cells are going to be B cells, and some of those B cells then are going to start secreting antibody, but now they're going to be secreting antibody of the donor allotype. So by typing the donor and the recipient for the allotypes, even though you don't necessarily have to match them for that, you can then, whether or not the bone marrow graft is taking if you can start detecting donor allotypes being expressed in the patient's serum. You know that the bone marrow graft took, and it's actually the cells have differentiated into B cells that are secreting antibody. Now, obviously, that's not the only thing you do in monitoring these patients, but it's one of the things that is used to see whether or not the grafts are taking, is by looking to see if the donor allotypes are being expressed in the recipient. Another place it's used is in forensic medicine. The allotypes can actually be detected in semen, in dried blood, in saliva, and so there's application in the forensic field for using allotypes to help identify people. And not so much nowadays anymore, because now, of course, we've got the DNA technology where we can identify, whether or not a person is the father of a child. But in paternity testing, it had been used a lot, and to some extent it is still used, because by these tests you can't prove, when you're looking at allotypes, you can't prove paternity. You can only disprove it. right? You can never prove that a guy is the father. You can disprove a guy is. So, and you can use the allotypes, and again, from the use of the genetics. If you had a situation where, let's say that the child is KM1, KM1, or I'm sorry, is a heterozygote. Let's say it's KM1, KM3, and the mother is KM1, KM1. If the guy, the putative father, is KM1, KM1, you know that that can't be the father, all right? So in paternity testing, then you can use these allotypes, at least to disprove paternity. Nowadays, of course, the DNA technology can, with a high degree of certainty, say that this is the father, so this stuff is certainly being replaced. But there's still some application of the allotypes in that field as well. All right, any questions about isotypes or allotypes? And I don't want to go on because those are a little bit easier. This is the difficult one. Okay? So if isotypes and allotypes are not clear, now's the time to ask the questions. I mean, obviously you can come later too, but if you've got them, let's deal with them now before we go on.
When you're talking about cells and how they can only express one allele, that is per individual B cell. Like you can have like one B cell with this one and one B cell.
Exactly, and that's why in the population of molecules, some of them have one and some of them have three, KM1, KM3, because some B cells excluded the 3 allele, another B cell will exclude the 1 allele, and so you get both different immunoglobulins in the population of molecules. Absolutely. Any other questions? Yes. Is it? Oh no, it's kind of, I mean, if we were to look on this here, we'd see a bunch of these different. I mean, that table I gave you, it was just a small number. I mean, there's a whole slew of these allotypes, okay? And I can guarantee you, we probably would not find two people that have exactly the same allotypes in this room if you looked at all of them. Now that we share a lot of them, obviously, but if you looked at the whole group, it would, you probably would never find anybody with exactly the same allotypes. No. These are all normal situations. Okay? Anything else? Yes.
When you were talking about like the nomenclature of the allotypes,
Right.
when it's like H1, what did you say the numbers were for?
It just specified which allotype you're talking about. That's allotype 1. KM1 would be the allotype 1 on the kappa light chains. It's just,
The order they found?
yeah, it's the order they found them usually is what it goes. Yeah, that was probably the first allotype found on the kappa light chain and so on. Okay? All right. Idiotypes. Again, our definition. Unique antigenic determinants present on individual antibody molecules or molecules of identical specificity. So what does it mean? Again, I want to do this one by a little experiment, so close your laptops. All right. Suppose I were to take a haptene carrier conjugate, DMP-BSA. DMP is the haptene. It's dinitrophenol, just a small molecule, okay? The carrier is BSA. It's bovine serum albumin. It's just a big protein, an immunogenic protein. And I make a haptene carrier conjugate. So I chemically coat the DMP to the BSA. And I inject this into a mouse of a strain A. Now, I need to tell you a little bit about mice strain. Laboratory mice have been inbred by brother and sister mating for 20, 30, 40 generations. And so they're highly, highly inbred strains of mice, okay? And so they've been inbred so much that essentially every mouse is genetically identical. They're all identical twins for all practical purposes, so genetically, okay? So we're going to take one of these strains of mouse, so we'll call it mouse strain A, and we're going to inject our haptene carrier conjugate into the mouse. Well, the mouse is obviously going to respond, right? And the mouse will make antibodies against the BSA, the carrier determinants, But in addition, it's going to make antibodies against the haptenic determinants, right? And those antibodies against the haptenic determinants will react with the free haptene, right? That's what we said earlier. So we have in this population of molecules, some of those are going to be anti-DMP antibodies. In other words, antibodies that will react with the free haptene. Some of them will be. All right. Now, again, if you would give me that it's possible for me to do this without me telling you how at this point, suppose it were possible for me to isolate those anti-DMP antibodies so that I could pull out those antibodies and only the anti-DMP antibodies, all right, and get rid of all the rest of them. Now I have purified anti-DMP antibodies. And I inject those antibodies into a strain A mouse. Not the same mouse up here, a different individual, but of the same strain. Okay? What will I get? Would you expect this mouse to make antibodies to any isotypic determinants that are present on those anti-DMP antibodies? Would you expect the mouse to make antibodies to any of the isotypic determinants? No. Why? There are no differences. I remember isotypes are present on all members of the species. So you're not going to get any antibodies against the isotypes. Would you expect them to produce any antibodies against any allotypic determinants on those? Why? No. They're genetically identical. There are no allotypic differences. Okay? So you won't get any anti-allotype. You won't get any anti-isotypic antibodies. Is there anything special about these antibodies that this mouse might be able to make antibodies against? The idiotype, right.
Their antibody, the variable things would be specific for something that they've never encountered, so they wouldn't have any.
Exactly. The combining site of this antibody that allows it to bind DMP is very, very unique, right? It's a particular set of hypervariable regions that create the combining site. So the combining site of this antibody is different. This mouse would see that combining site and make antibodies against the combining site of the anti-DMP antibodies. Well, what makes up the combining site? The hypervariable regions, right? So it's the antigenic determinants that are created by the hypervariable regions of the immunoglobulins that are called the idiotypes. Okay? All right, does that make sense? When any antibody that, in this case, has specificity against DMP, has a very unique set of hypervariable regions that allows it to make a combining site that will bind DMP. And so those hypervariable regions then can be an antigenic determinant that this mouse recognizes and starts making antibodies against. So this is antibodies against the combining site. And more specifically, they're antibodies against the hypervariable regions. Now, the actual determinant, the antigenic determinant, although it's certainly influenced by the hypervariable regions, may extend somewhat into the framework regions as well, but it's really dictated by those hypervariable regions. And, of course, the hypervariable regions are found on both the heavy and the light chains. And so there can be idiotypic determinants on both heavy chains and light chains because they both have hypervariable regions. Okay? Some of you are looking kind of shell-shocked. Yes.
So the first mouse that produced that antibody doesn't make the antibody against the antibody?
I'll come to that in a second. Good question. I'll come to that in a second. All right. Is it clear what the idiotypes are? All right. So if these are determinants that are created by the hypervariable regions. Now, as I said, you can find them on both the heavy chain and the light chain. Sometimes, however, the actual determinant becomes a conformational determinant that is created by both the hypervariable region of the light chain and the heavy chain. So in other words, it's some conformation that involves both hypervariable of the light and hypervariable of the heavy. In those instances, obviously, if you separate the heavy and light chain, you destroy the epitope. So it's if a conformational determinant, you destroy the epitope. But in other instances, it's directed only against say, the variable region of the heavy chain or only against the variable region of the light chain. In that case, you would not destroy the epitope by separating the chains. So you will find both conformational and sequence determinants in these as well. Okay? All right, is it clear what the idiotype is? It's an antigenic determinant that's created by the hypervariable region. Every antibody, then, of different specificity has a different idiotype. Because the specificity is determined by the hypervariable regions, so every different specificity represents a different idiotype. All right? Okay, so where are they going to be located on the immunoglobulin molecule? Well, obviously, they're going to have to be in the variable region, right? Because that's where the hypervariable regions are found. They're going to be up in the variable region of the heavy chain or the variable region of the light chain is where you're going to find the idiotypic determinants. So let's look at a situation here. We have two IgG1 kappas from the same person now. But one of them is against antigen A, and another one is against antigen B. These immunoglobulins will have the same isotypes indicated in yellow, right, on both the heavy chain and the light chain. They will have the same allotypes. They're from the same person on the heavy chain, the same allotypes on the light chain. But they will have different idiotypes, okay? This has one set of idiotypes. This has a different set of idiotypes because they have different combining sites. So every antibody with the different specificity has a different idiotype. Okay, so what are they used for? What's the usefulness of the allotypes? Well, immunologists like them because now we finally have a marker for the variable region. All the other things were for the constant region, but that's not important for you. One place where it is important is that they're involved in regulating immune responses. And let me illustrate what I mean by that. And this speaks to your question, okay? When I take an antigen and then immunize somebody with an antigen, it leads to the production of antibody. But for our purposes, I'm going to call this idiotype, right? Because the antibodies have a particular idiotype. So let's call it idiotype. So we're going to get the production of some idiotype, okay? Because the antibodies will have a particular combining site. All right. Now, once this is made, and this is the question that you had, once this is made, that's something that we've never seen before. So what happens is that the presence of this leads to the production of another antibody directed against the idiotype. In other words, an anti-idiotype. Now, it turns out that if you look at these, if you analyze these anti-idiotypic antibodies, they actually fall into two types. There are some of them that will actually stimulate the production of more idiotype, and some that will inhibit the production of idiotype. So what really controls then the amount of idiotype antibody that we make is three things. It's the amount of antigen that's there driving this production of this, as well as the ratio of the positively regulating and negatively regulating anti-idiotypes. So what controls this is all three of these. Is antigen there driving it this way? And then do we have anti-idiotype positively and negatively? What's the ratio of these two that regulate this? So the concentration of this is regulated by antigen as well as anti-idiotype. Okay? But this is just an antibody, right? So it's got an idiotype. So that leads to the production of an anti-idiotype, which can be positively or negatively regulating. So what regulates the level of the anti-idiotype is the presence of the idiotype as well as the presence of the anti-idiotype, then positively and negatively regulating, right? But that's an antibody, and it's got an idiotype. which leads to the production of an anti-idiotype, and so on. So you get the picture. So you have this network of idiotype, anti-idiotypes interactions that help regulate these immune responses and monitor and keep the levels of these antibodies present, okay? So this is referred to as the regulation by the idiotype, anti-idiotype network. And in fact, you can actually, it's been demonstrated experimentally. For example, if you inject an animal with an antigen, you obviously can detect the idiotype, which is the antibody. You can actually detect the anti-idiotype, the anti-anti-idiotype, and the anti-anti-anti-idiotype. They've gone back experimentally and shown 3 levels back that you can actually detect these different antibodies actually being made in the individual. Yes? Well, obviously, if you don't want to just keep making antibody, if you just kept making antibody all the time, it would be no use. Once the antigen is gone, you don't need it anymore. So you want to control it then. So this just helps control the level. So what drives it is, as long as antigen is present, we're getting it. Afterwards, we can maintain some level of the thing by these as well. Okay? Yeah. No, in fact, the reason I didn't want to confuse, this gets to be kind of hard to conceptualize. So I put it in a separate mouse just to make it a little bit easier. But no, exactly the same thing would have happened in in the first mouse that got injected. Okay. Yes, the anti-idiotype is something against the hypervariable region of this antibody molecule. And this one is an antibody against the variable region combining site of this antibody. Okay? Okay? No, it has got a combining site against it is an antibody against the combining site present on these antibodies. And this has an antibody against this one. Okay? Say that again? Yes, right. This is the only thing that will bind to this. Right. This is the only thing that will bind with antigen. Okay? All right. Another place where idiotypes, there's a lot of interest in idiotypes, is in the vaccine area, to try to develop what I refer to as anti-idiotype vaccines. So how can you use an anti-idiotype as a vaccine? Well, let's go back to this. We saw what happened when we injected antigen. We get this production. We get these positively and negatively regulating anti-idiotypes. If it were possible to selectively pull out these anti-idiotypes that positively regulated the idiotype, okay, if you could do that, and that is obviously the big trick in this. If you could do that, then to produce this antibody, you wouldn't need antigen anymore. That's all you'd need is the anti-idiotype. You can inject the anti-idiotype, and you would get the production of the antibody. So, I mean, why would you want to do this? Why not just inject antigen? Well, if the antigen is particularly toxic or dangerous to administer, and you don't want to do this, this is an alternative approach, okay, to inject the anti-idiotype. So there's a lot of interest in trying to develop these anti-idiotype vaccines as a way of getting around the fact that we can't always inject the antigen because it may be too toxic. Okay? Another place where idiotypes and anti-idiotypes are used is in the treatment of B-cell tumors. Remember we said B cells have on their surface immunoglobulin, IgM and IgD. They have the same specificity, so they have a particular set of hypervariable regions. That means that those B cells, every B cell expresses a particular idiotype, depending upon whatever the combining site of those antibodies are. Now what happens when you have a B-cell tumor? You get a malignant proliferation of a clone of cells. So that clone of cells starts dividing, and you have lots of these cells, right? And then you have a malignant proliferation of these cells. So all the cells in the tumor that are tumor cells will be expressing the same idiotype. So if you could produce an antibody, an anti-idiotype, to that idiotype that's present on the tumor cell, then you can treat the patient with that. You can administer the anti-idiotype. The anti-idiotype will very specifically bind only to the tumor B cells, sparing all the normal B cells. It's highly targeted to that, and it can kill the tumors. It can kill the tumors maybe because it fixes, the antibody fixed complement and relies on that, or what's more typically done is it's possible to couple to the antibody molecules toxic materials that then kill the cell. So you use the anti-idiotype to target specifically the tumors, and then you kill only the tumor cells, sparing all the normal cells. Yes? And that would happen. That would certainly happen. Or it could happen. All right. But you treat and if you kill off all the tumor cells, you've treated the patient at least, as long as you can get the cell. Yeah, obviously it's going to take some time for that to happen. So you've got some time that you can work, but ultimately that theoretically can happen.
Like each person that has a B-cell tumor, it's like you have to, as a physician, have to take part of that and figure out what the idiotype is.
Yeah, actually what they do, you have to take out some of the tumor cells, get the idiotype, and then you prepare the anti-idiotype from that, and then infuse it back into the patient. So yeah, obviously it's tailored therapy, because every tumor is going to have its own idiotype. Okay? Now, we haven't talked a lot about T cells, but I will tell you that the same thing applies to T cells, because T cells also have a receptor on their, for antigen on their surface, and those receptors also express an idiotype, because they have hypervariable regions as well. And so they express an idiotype. And so, T cells have idiotypes as well. So there's no reason that you couldn't produce an antibody, anti-idiotype against the T cell idiotypes and use those for the treatment of T cell tumors as well. Yes?
This is still in research?
Yeah, pretty much so. But it's clearly, the direction that people are heading. They're trying to do these kind of things.
Have one noticed that there's type with among the population that happens to cause cancer?
Not that I'm aware of. You're looking for a general reagent that could be used and then to treat. Not that I'm aware of. All right. Any questions on allotypes, idiotypes, or isotypes? Yes.
I don't really understand how we can create antibodies to something in our own body. We say it's a new antibody that we're producing, but really all our antibodies are based on their names of genetic origin. And theoretically, I've got a B cell somewhere that's already presenting that antibody, right?
Right, but they're there at such low concentrations because they're only on the surface of the B cells.
So is it?
In other words,
In other words, that B cell would be activated and changed, and you'd have plasma cells making that antibody, and then some other B cell would the antibody for the one that you just made.
right. Then it would act. All right. All right. All right. What we want to talk about now is a little bit about the genetics of the immunoglobulins. And the reason that we incorporate a part on the immunogenetics, or the genetics of the immunoglobulin molecules, is because the genetics really has explained a lot of the phenomenology. I've been telling you a lot of things. I've been telling you things that B cells have on their surface, both IgM and IgD, but yet the specificity of those, that IgM and IgD is the same. I told you that there's this thing called allelic exclusion, but how does all that happen? And many of these questions really were answered from an understanding of what's going on at the level of the genes for the immunoglobulin molecules. And so many of the things that we've talked about will be explained by the genetics. And so we're going to cover the genetics of the immunoglobulins. And let me just sort of give you a little historical perspective for the immunoglobulin genetic story. When all this sequencing was going on, where people were sequencing the heavy chains and the light chains, one thing became very clear from these studies. The same constant region could be associated with many variable regions. So, for example, you could find IgG antibodies that had a lot of different specificities. All right? So the same constant region that determined a gamma 1 or a gamma 2 or a gamma 3 could be associated with a lot of different antibody specificities. But the sequence studies also showed that the same, if it'll let me, the same variable region could actually be associated with many different constant regions. So, for example, you could find an IgM and an IgG molecule that had the same idiotype. And the idiotype, remember, is a variable region marker, so it meant that they had the same variable region. So here you have the same variable region on two different classes of immunoglobulins. So the question can be asked, well, how can you have a situation like that? How can you have the same constant region associated with a lot of variable regions, or the same variable region associated with a lot of different constant regions? And in an attempt to answer that question, the immunologists suggested at the time that maybe what's happening is that you have two genes that encode the immunoglobulin molecules, a variable region gene and a constant region gene. And you could then just sort of mix and match which genes you're going to put together. You could take this same variable region and put it with a bunch of constant regions, or vice versa. So they suggest, well, there's two genes coding for this protein. Well, obviously it would be easy to explain how you get these results with that. But the problem was, at the time that this suggestion was made, and we're talking about this is going on in the late 60s, early 70s, probably before all of you were born. But that's the time that roughly, late 60s, early 70s. The problem is that our understanding, or our concept of gene at that time was one gene, one polypeptide. And here these immunologists are saying two genes, one polypeptide. Didn't make any sense. Okay, nobody wanted to accept it. So what happened was that people said, well, no, all right, we got to have two genes. That's the easiest explanation. But then what happens? So each gene makes their protein, because now we got to obey the rule: one gene, one polypeptide. Okay, so now we make two proteins and we stick the proteins together somehow. Somehow you then form a peptide bond between the variable region protein and the constant region protein, and that would do it. Okay, so people then looked for evidence, that the two proteins were coming together. Nobody could find any evidence. In fact, there were some really exquisite experiments done to show that that doesn't happen. There is no fusion at the protein level. So then people say, well, okay, let's make the fusion at the RNA level. So you have two genes, they make two messages, we link the messages together, and then we translate it into one protein. So then they looked for evidence for that. Nobody could find it, all right? And in fact, the evidence said absolutely not, that doesn't happen. It wasn't until the mid-70s that the first evidence came out that something was happening at the DNA level. That in fact there are two genes that they are not separated. They happen to be on the same chromosome, but they're not the same gene. They're separated on the chromosome, and some rearrangement occurs at the DNA level. And in fact, we know now that in fact the Immunoglobulins are coded for by 3 gene families. One gene family encodes the kappa chains, kappa light chains. Another gene family encodes the lambda light chains. And the third gene family encodes the heavy chain genes. And all of these gene families are actually on separate chromosomes. All right, so we have distinct groups of genes that encode these 3 different classes. And what we want to do is then to look at these gene families and look at how they're expressed. So what we're going to do is we're going to look at the way the genes are organized in the germline in a cell that is not differentiated in any way to be a B cell. So this may be a stem cell, or actually it could be any cell. It could be a kidney cell or a liver cell, whatever, anything that is not a B cell. We're going to look at the structure, the organization of those genes, and then we're going to look to see what happens when a B cell starts undergoing B cell differentiation. What happens to those genes? All right? So we'll start with the lambda chain, light chains. This is the gene organization in the germline, in the undifferentiated cells. In the case of lambda, there is actually one constant region gene for every subtype of lambda. So we have 4 constant region genes in this gene family. The one codes for the lambda 1, lambda 2, lambda 3, and lambda 4. And by the way, in all of these in subsequent figures, the boxes are going to represent the exons, the coding sequences. The thin lines in between them are introns. This is the intervening sequences of DNA. All right, so we have there's one exon for each type or subtype of light chain, of lambda light chain. Now, quite a ways away from the constant region genes, and that's why I put a dotted line in here. We're talking about, this is a huge piece of DNA actually, but quite a ways away, you find the variable regions. And there are multiple variable regions. In the case of lambda, there's about 30 different variable region genes that encode lambda variable regions. And each of these genes has this. There's the main exon that codes for the bulk of the gene. Then there's a short intron in here, and then there's a short sequence labeled L that stands for leader region, and we'll see the importance of the leader region shortly. But each one of these variable regions has a leader region associated with it. There's one additional coding sequence in here, which is indicated here in white, and upstream of each of the constant region genes, there's this other J gene segment. And we typically don't refer to it as a gene. We usually just call it a gene segment because it really only codes for just a few amino acids. It's just, 5 or 6 amino acids encoding. So it's just a piece of a gene, basically. So the upstream of each of these, we have a J gene segment. And it's unfortunate, the nomenclature. Don't get confused. That J has nothing to do with the J chain that you find in IgM or anything. Okay, so it's totally different. It's just the J stands for joining. Okay. All right. So that's the organization of the lambda light chain genes in the germline. What about kappa? Kappas look like this. You have a single kappa heavy constant region gene. There's only one type of kappa chain. There's only one kappa constant region gene. You also have multiple variable region genes separated quite a distance on the chromosome from the kappa gene, from the C-kappa gene. Right? And each of these variable regions have a leader region associated with them. And in the case of kappa, there's about 300, roughly in that ballpark. Those are the estimates, the number of genes. And you also have J gene segments. But in the case of kappa, you have multiple J gene segments upstream of the C-kappa gene. Right? So that's the way the gene looks in its germline configuration. What we want to do now is to look at what happens when a cell in the bone marrow, when a precursor of a B cell, a stem cell in the bone marrow, commits to becoming a B cell and undergoes B cell differentiation. What happens to these genes? And we'll follow the example with the kappa. The same is going to be true of the lambda, so I'll just take you through with kappa. So here's our germline organization with kappa. All right? So what happens? What happens is that there's a DNA rearrangement. A recombination event occurs between one of these 300 variable region genes and one of these 4 or 5 J gene segments. A rearrangement occurs. In our example here, a rearrangement is going to occur between V kappa 2 and J kappa 4. It should be a 4. All right? So between J kappa 4 and V kappa 2. This is a recombination event that occurs. And what happens is all this DNA in between here gets deleted and is removed. So that we now bring V kappa 2 in our example, now immediately adjacent to J kappa 4. Now, it is a, quotes, random event as to which V gene is used and which J gene segment is used. And the reason I put random in quotes is because there does seem to be some bias for choosing those variable region genes that are closest to the J regions. So in other words, this gene here has a greater probability of recombining than this gene over here. But eventually, in the B cell population, there's going to be a B cell that will recombine every one of these things to one of the J gene segments. So there's one B cell is going to take J2 next to V2 next to J1. Another B cell may take V2 next to J2. Some B cells are going to take V1 next to J1 and so on. All possible combinations will eventually be expressed, although there does seem to be some preference. The number of cells that use this one to recombine is a little bit higher, okay? But so that's why I put random in quotes. But eventually it's random and all possible combinations are made. All right. So now we've rearranged the DNA, and all that DNA in between here is removed from the chromosome and is actually deleted out. All right? Now, the consequence of this rearrangement at the DNA level is that the gene, these genes now become transcriptionally active. And the reason they become transcriptionally active is the following. Here's up front of each of these in the variable region gene up in the genome, in the germline, you have a promoter that drives the expression of these genes. But the promoter is not a particularly good promoter, and you don't get transcription initiating from these things normally. It requires some help, and it requires the help of an enhancer. Now, I assume you had enhancers in biochemistry? All right, so it requires an enhancer in order to get good transcription initiating at that promoter. There is an enhancer in the system, but it's sitting here, right there, right upstream of C kappa. And if you remember the properties of enhancers, they can enhance transcription from promoters, but one of the requirements is that they have to be within a certain distance of the promoter. Well, in the germline, this enhancer is sitting far away from any of these promoters, and so it can't enhance transcription, and that's why you don't get transcription of these genes. After the recombination event, however, this enhancer is now brought close enough to the promoter in the rearranged gene so that it now can initiate transcription from this promoter. It's still far enough away from all these other promoters it doesn't initiate transcription from those other promoters. It only initiates transcription from this, because this is the only one that's close enough now. So we now start transcribing this gene. And when we get the transcript starts here. We transcript the leader, the V, the J, and so on. And you go here. And the end of transcription is at the end of the C kappa gene. And you make a primary transcript that looks like that. All right? This transcript then is processed or spliced, whatever you want to call it. It's RNA processing, RNA splicing. And the processing is in such a way is that this intron is removed and this whole area is removed. And we end up then with the messenger RNA for a kappa light chain. And the messenger now has all the coding sequences adjacent to each other. All right? This message then gets translated into protein. The leader segment, the leader region here, has got a signal. It contains a signal that says transport me to the lumen of the endoplasmic reticulum. So As this protein is being made, the leader directs the protein to go into the ER, and you end up with the protein in the ER. In the process of transport into the ER, the leader region is actually clipped off, and you end up in the lumen of the endoplasmic reticulum with a kappa light chain. What we had referred earlier to as the variable region is actually encoded for by the variable region gene as well as the J gene segment that was used. So the variable region is encoded by V and by J. What we had referred to as the constant region is encoded for by the constant region gene in the germline. Okay? And so now we have made a kappa light chain, and that's in the lumen of the ER. When this cell makes a heavy chain, then that'll assemble with the light chain. Okay? The heavy chain will assemble with that light chain, will make an immunoglobulin molecule, ship it off to the surface of the cell to be displayed on the surface of the cell. Okay? Any questions? All right, we'll stop here and pick up the heavy chain genes tomorrow.
Questions that I get confused easily. So I was wondering, in this figure, it's a little bit different from what you had up on the screen. And I was wondering, are these supposed to be kappas here? Because
yeah,
we have kappas.
I never realized that.
Okay, good. So that helps.
Yeah.
All right. And then I had asked you about the roughly 50-50. And I just did a Punnett square, and I came up with 75-25. Is that roughly 50-50 to you? That's why I was...
No.
So if I have 1-3 and 1-1, so
you're going to get
1-1. So 1-1, 1-1, 1-3.
Yeah, you're right. 1-1, 1-1. No, 1-1, 1-1. Cam.
The 1-1 is going to dominate because.
No. You're going to get 1-1. If the mother gives this and the father gives this, or the mother gives this, the father gives this, we have 1-1, 1-1. Alternatively, the mother gives this, the father gives this. Or the mother gives this, father. So you're going to have half of them are going to be 1-3, and half of them are going to be 1-1.
I'm sure you know more about this than I do. That's why I'm confused. It's just I always do this with a Punnett square, and I always end up with you just take the alleles and put them there, and so you end up with 75 because you're just you're in the pool. You're just much more likely to get a 1-1. Oh no, it is a 3. You're right.
Yeah,
3 goes here.
yeah, and there's something. There has to be something wrong.
Yeah, I knew I was wrong. I knew you were right.
There has to be something wrong. I know it's 50-50.
Yeah, because this guy can only give 1 or 3.
Exactly.
Okay. Thank you.
Okay.
I still haven't been able to get the font for the Greek letters.
They didn't load?
I tried with the email to load them on my computer, and it just didn't do anything.
I wonder if anybody, nobody else has said that they've had problems with it, so this is the first one. I wonder if...
I've just been going in and changing all the little characters.
Yeah, let me get with, let me talk to Sean about it and see, because he thought he had gotten the right fonts.
Okay.
Because we had this problem last year, and that's when he gave them, and when they loaded that on.
Okay.
You know who Sean, tomorrow in class, when he comes here early. Let's talk to him. He's the guy that comes here and gets that. So that's okay. Sean. Okay. So when he's here, stop up and let's talk to him. Okay.
Hi there.
I'm Rick Forrest.
How you doing?
I'm the class president. I just want to make a quick announcement.
Oh, please do. Yeah.
When it starts quieting down,